Yes. Now we will see regarding this gluconeogenesis, how this is going to happen. So the once after the pyruvate molecule, which we have seen all the fates in the last PPT. So now this synthesis of glucose, once after the formation of pyruvate molecule, now again we will see how the glucose is going to be synthesized. What is the need of this synthesis of glucose process? That is nothing but the gluconeogenesis process through the gluconeogenesis. So the gluconeogenesis, how you are going to define? First of all, it is a process of converting non-carbohydrate precursors to glucose or the glycogen molecule. So this gluconeogenesis, which is going to meet the needs of the body for the glucose when sufficient carbohydrate is not available. When sufficient carbohydrate is not available from the diet or the glycogen reserves. So then this gluconeogenesis is going to happen. So it is nothing but where you can come across the supply of glucose which is necessary especially in the nervous system, and as especially for the nervous system and the erythrocytes. So if the gluconeogenesis is going to be uh, fails, then that is usually fatal. During the fatal condition, so this gluconeogenesis is going to be not happening, or the failure of the gluconeogenesis which we can come across. So what is the need of this synthesis of glucose? Why we have to synthesize the glucose molecule? Mainly, first of all, what is the need of this is nothing but to maintain the blood glucose level. So within a narrow range, we have to maintain the blood glucose level, that is 80 to 120 mg per deciliter. So in order to maintenance of this, we have to go for this gluconeogenesis process, or we have to go for the synthesis of glucose through gluconeogenesis process. So what are all the tissues which is involved in this? So brain, erythrocytes, testes, and the kidney medulla are mainly dependent upon this glucose molecule for the continuous supply of energy. So this particular tissues, along with that tissues, this skeletal muscle, during exertion condition, that is anaerobic condition, it is using glucose at a rapid rate. So as well as in the fasting condition or the starvation condition, in order to meet the basal requirements of the body for the glucose, is essential, maintain the requirements of the body for the glucose. So it is mainly required, and it is also acting as a very essential source for the survival purpose, as well as prevent the it effectively prevents the accumulation of the certain metabolites in the blood, that is nothing but lactate molecule or lactic acid, as well as glycerol and the propionate. So these are all the things. So we have to require, especially in order to maintain this, we have to synthesize the glucose molecule through the gluconeogenesis. First of all, we will see what are all the substrates which are required mainly for the gluconeogenesis process to carry out. So the major substrates, which are the gluconogenic amino acids, lactate molecule, glycerol, and the propionate molecule. So these non-carbohydrate precursors, these are considered as a non-carbohydrate precursors of the glucose, which are first converted into pyruvate molecule, or it is going to enter into the pathway at the later as a later intermediates such as oxaloacetate and as well as dihydroxyacetone phosphate, and enter into the glycolysis. So this particular process you can see here in the case of liver and kidney, which are going to be acting as a major tissues for this gluconeogenesis process to carry out. So first substrate molecule, that is glucogenic amino acids. So these amino acids, which are going to be derived from the dietary proteins or the tissue proteins, or even from the breakdown of the skeletal muscle proteins during starvation condition or during strenuous exercise or the intense activity of the skeletal muscle. So after transamination or the deamination of the particular transamination reaction, that is nothing but the transfer of amino group, or the deamination, that is nothing but the removal of amino group, this particular glucogenic amino acids, which are going to yield either pyruvate molecule or the intermediates of the citric acid cycle. So which are the intermediates of citric acid cycle? Maybe it is yielding as a succinyl CoA, or it may be yielding as a alpha ketoglutarate, or even it may be yielding as a malate, or even it may be yielding as a oxaloacetate, as well as mm, uh, oxalostate, etc. So these are all the intermediates of citric acid cycle which are going to be formed after transamination or this deamination reactions. Coming for the next substrate molecule which is very important and playing a role in gluconeogenesis process, that is a lactate molecule. So this lactate is going to be formed by the active skeletal muscles when the rate of the glycolysis which exceeds the rate of oxidation process or the oxidative metabolism. When the rate of oxidative metabolism is going to exceed, the lactate is going to be formed by the active skeletal muscle. So you can see here the process of formation of lactate from the from the pyruvate molecule, lactate is going to be produced. So this lactate is readily converted into pyruvate by the action of the enzyme lactate dehydrogenase. So here we have seen in the fates of pyruvate is getting converted to lactate, that is acted upon by the enzyme, same enzyme, that is LDH, that is lactate dehydrogenase, that is a reversible reaction as we have seen. So there it was utilized NADH NAD plus which was utilized, and it is forming it was utilized NADH molecule and regenerating NAD plus molecule. That NADH which was utilized from the glycolysis path, you have remembered, right? So that NAD plus which is generated is utilized here also you can utilize that is lactate to pyruvate conversion. So this NAD plus is utilized in the lactate formation of pyruvate from the lactate molecule which is acted upon by the same enzyme lactate dehydrogenase. This shows the reversible reaction of this. So coming for the biological significance of this LDH, what is this LDH significance in the biological way? So first of all, in the direction of this lactate formation, the LDH reaction which requires NADH and yields NAD plus, as I said you just now, which is then available for the use by the glycerolidate phosphate dehydrogenase reaction, which is done in glycolysis process or acted upon in this enzyme through aerobic glycolysis. These two reactions are therefore you are going to intimately couple during anaerobic glycolysis, that is pyruvate to lactate formation and lactate to pyruvate formation. So these two reactions can be coupled here because of the utilization and generation of NAD plus and again regenerating NAD plus molecule and utilizing here and forming NADH. So secondly, the lactate produced by the LDH reaction is going to be released into the bloodstream and then transported to the liver where it is getting converted to the glucose molecule. Then this glucose is further returning to the blood and it is used up by the muscles as an energy source, and it is going to replenish the glycogen stores, and it is going to replenish as a glycogen stores. So this cycle is termed as Cori cycle. I'll repeat it once again. So what is going to happen here? The lactate which is produced in the LDH reaction is released into the blood and bloodstream, and it is going to be transported to the liver. Then it is going to be converted to the glucose there. The glucose which is then further transported to the blood for the use by the muscle as an energy source, and it is going to replenish as the glycogen stores. So this particular cycle is termed as Cori cycle, blood to liver, liver to further back to blood. So it is termed as Cori cycle. Coming for the next substrate molecule which is utilized in this gluconeogenesis process, that is a propionate. So this propionate is acting as a major source of the precursor of the glucose molecule in case of ruminants, where it enters into the gluconeogenesis process via the citric acid cycle pathway, or even it is also called as TCA cycle. So once after the esterification with the coenzyme A, propionyl CoA is going to be carboxylated further to form D-methylmalonyl CoA. So this formed D-methylmalonyl CoA, which is catalyzed by the enzyme propionyl CoA carboxylase, a biotin dependent enzyme. So this D-methylmalonyl CoA further undergoing racemase reaction. So this methylmalonyl CoA racemase is going to catalyze the conversion of D-methylmalonyl CoA to L-methylmalonyl CoA, then further undergoes isomerization reaction to form succinyl CoA, which is further catalyzed by methylmalonyl CoA mutase reaction. You know the racemase and isomerase and the mutase differences, right? Racemization is happening, racemic mixture is going to be formed there. So here you can see conversion of dimethyl CoA to L-methyl CoA, that is racemase acted upon by racemase, further acted upon by isomerization. That is isomer of this methyl CoA is succinyl CoA. So then this succinyl CoA is further catalyzed by the enzyme mutase. So mutase reaction is happening, that is succinyl CoA to methyl CoA. So further this methyl CoA mutase is going to be acted upon by this succinyl CoA to form methylmalonyl CoA. Then this particular methylmalonyl CoA enzyme is an biotin dependent enzyme, B12 vitamin B12 sorry, it is a vitamin B12 dependent enzyme. If this deficiency of this enzyme leads to further accumulation of this methylmalonic acid, which cannot be further converted to particular metabolite, so it is going to be further accumulated in the blood and is excreted in the urine. This particular disorder is called as methylmalonic aciduria, because of the accumulation of methylmalonic acid in the blood, and it is going to be excreted in the urine. Coming for the next molecule, that is next substrate, that is the glycerol. So this hydrolysis of this triacyl glycerol, so as you all know, in the fat cells which are going to yield the glycerol and fatty acids. So this glycerol may enter into the either the gluconeogenesis process or the glycolytic pathway as a dihydroxyacetone phosphate formation. So in the fasting condition, you can see in the fasting state, the glycerol is going to be released from the lipolysis of the adipose tissue. Triacyl glycerol is mainly used solely as a substrate for the gluconeogenesis process in the liver and the kidney. So this requires phosphorylation first. So this requires first of all the phosphorylation reaction of this glycerol, which is acted upon by the enzyme glycerol kinase to form glycerol 3 phosphate. So then this glycerol phosphate is going to be acted upon by the enzyme dehydrogenase, that is undergoing dehydrogenation reaction to form glycerol 3 phosphate dihydroxyacetone phosphate. You can see this dehydrogenation reaction which is happening here. So you can see here to form dihydroxyacetone phosphate, which is catalyzed by glycerol 3 phosphate dehydrogenase. So once kinase reaction is happening, you can see here in this slide glycerol phosphate is going to be formed. You can see the phosphate group which is going to be at the third carbon atom of this glycerol. So what is the structure of glycerol? As you all know, CH2OH, CHOH, CH2OH. So it is undergoing phosphorylation first, acted upon by glycerol kinase to form glycerol 3 phosphate. Third carbon atom is occupying the phosphate group. So next it is acted upon by glycerol phosphate dehydrogenase. Dehydrogenation reaction is happening. So NAD plus is the cofactor. You can see here formation of NADH. So dihydroxyacetone phosphate. Now this dihydroxyacetone phosphate which is formed, it can continue the process of gluconeogenesis in the reversible reactions, reversible way. So coming for the reactions of gluconeogenesis, first of all, this gluconeogenesis is not the exact reversal of glycolysis. It is just a three reactions which are going to be bypassed, three reactions which are going to be bypassed in the gluconeogenesis process. Three reactions of this glycolysis which are going to be bypassed in the gluconeogenesis process, where you can come across, except those three reactions, other reactions of gluconeogenesis is exactly reversal of glycolysis. So you can see here in the gluconeogenesis, what are all the new steps which are going to be bypassed this particular irreversible reactions of glycolysis, which we have seen in the glycolysis pathway. So first bypass reaction is formation of phosphoenol pyruvate from pyruvate molecule. So this is the first bypass reaction, which is catalyzed by the enzyme pyruvate carboxylase, combined action of pyruvate carboxylase and phosphoenol pyruvate carboxykinase. So combined action which we are going to see, that is pyruvate carboxylase and phosphoenol pyruvate carboxykinase. Second bypass reaction is nothing but formation of fructose 6 phosphate from fructose 1,6 bisphosphate. Third bypass reaction is nothing but formation of glucose by the hydrolysis of glucose 6 phosphate. So we will see first one by one. First bypass formation of phosphoenol pyruvate from pyruvate molecule. So what is going to happen here? So first this phosphoenol pyruvate, which is formed from the pyruvate molecule by the way of oxaloacetate through the action of this enzyme, that is pyruvate carboxylase and phosphoenol pyruvate carboxykinase. That is a combined action of this enzyme pyruvate carboxylase and phosphoenol pyruvate carboxykinase, which catalyzes this reaction to happen, that is formation of phosphoenol pyruvate from oxaloacetate. First pyruvate to oxaloacetate is going to be formed, then oxaloacetate to phosphoenol pyruvate is going to be formed by the action of phosphoenol pyruvate carboxykinase. So first pyruvate carboxylase is it is first of all the mitochondrial enzyme. It is going to happen in the mitochondria, this particular reaction, whereas the other enzymes of this gluconeogenesis are cytosolic in nature or cytoplasmic in nature. So they are going to happen in the cytosol itself. Only this reaction, that is pyruvate conversion of pyruvate to oxaloacetate, that is catalyzed by the enzyme pyruvate carboxylase. We can see in this slide. So the pyruvate carboxylase is catalyzing the carboxylation of this pyruvate molecule to oxaloacetate, which is an ATP requiring reaction. You can make out here. It is also a vitamin biotin dependent enzyme. So this biotin dependent coenzyme, so this enzyme requires this biotin molecules to carry out this carboxylation reaction. So first biotin is going to bind to the carbon dioxide, binds the carbon dioxide molecule from the bicarbonate ion. So here the carbon dioxide provider is bicarbonate ion. So it is going to form as a carboxybiotin, which is prior to the addition of the carbon dioxide to the pyruvate molecule. So it is going to form as a carboxybiotin before the addition of this carbon dioxide molecule to the pyruvate. You can see here two carboxyl group is seen in the formation of oxaloacetate. So we can see this is a pyruvate carboxylase reaction, which is acted upon by this enzyme pyruvate carboxylase, which is an ATP biotin dependent enzyme. So in the next we can see how this oxaloacetate is going to be transported. So this oxaloacetate, which is formed as the by the product of this action of the enzyme pyruvate carboxylase, so further reduced to malate. It is going to be reduced to malate inside the mitochondria, and it is going to be transported to the cytosol. First it is reduced to malate inside the mitochondria. It is first reduced to malate, then it is going to be transported to the cytosol. So this reduction is happened or accomplished by the NADH linked enzyme, that is called as malate dehydrogenase. So once this mitochondrial enzyme malate dehydrogenase, so it is occurring in two locations, that is mitochondrial malate dehydrogenase and cytosolic malate dehydrogenase. You have two location of this enzyme malate dehydrogenase. So first this is going to be reduced to malate in the mitochondria itself. That oxaloacetate is first reducing into malate by utilizing this mitochondrial malate dehydrogenase, which is an NADH linked enzyme. So once after the formation of this malate then this malate is going to be further transported across the mitochondria by using this malate shuttle, so which we will see in the later slide. So this malate has been transported across the mitochondrial membrane, and it is going to be further reoxidized to oxaloacetate by an NAD plus linked enzyme, that is malate dehydrogenase, which is going to happen in the cytosol, that is cytosolic malate dehydrogenase. As you clear now, understanding. So once the particular oxaloacetate, I'll repeat it. So once the first oxaloacetate is going to be formed, it is formed in the mitochondria. First it is reduced to malate in the mitochondria itself by acted upon by the mitochondrial malate dehydrogenase enzyme, which is nothing but NADH linked enzyme. So this is going to be further transported across the mitochondrial membrane, then it is going to be reoxidized to oxaloacetate by an NAD plus linked cytosolic malate dehydrogenase. Understanding? So cytosolic malate dehydrogenase. So once it is transported to the cytosol, it is acted upon by cytosolic malate dehydrogenase. So you can see here in this particular transportation in this particular slide how this is going to happen. So first this you can see cytosol, this is the mitochondrial matrix. So first the pyruvate is pyruvate is getting converted to oxaloacetate acted upon by pyruvate carboxylase enzyme, which is a biotin dependent. Then oxaloacetate is further converted to malate acted upon by mitochondrial malate dehydrogenase. Then this mitochondrial malate dehydrogenase, which is catalyzing once after the formation of malate is entering into the cytosol now. So this malate is entering into the cytosol This acted upon by transported across the mitochondrial matrix and entering into the cytosol. It is acted upon by cytosolic malate dehydrogenase now here to form oxaloacetate. Now this oxaloacetate is undergoing decarboxylation reaction. You can see here the second enzyme, that is phosphoenol pyruvate carboxykinase. This phosphoenol pyruvate carboxykinase, which catalyzes the decarboxylation reaction, and phosphorylation of this oxaloacetate is going to happen. Both decarboxylation and phosphorylation of oxaloacetate is going to happen. So here phosphoenol pyruvate is going to utilize GTP as the phosphate donor, not the ATP directly. It is utilizing GTP as the phosphate donor, so which we can come across here. Same thing what we have seen in case of what we are going to see in case of TCA cycle also. There also one enzyme, that is succinyl thiokinase enzyme, is utilizing GTP molecule instead of ATP directly. So the same thing which we are going to see here also, phosphoenol pyruvate carboxykinase is also utilizing GTP molecule as an energy molecule to carry out this reaction to form, that is decarboxylation of oxaloacetate to form phosphoenol pyruvate. So what is the significance of this decarboxylation of oxaloacetate? So in the liver and kidney, where the reaction of this succinate, just now that's what I said, succinate thiokinase, in the citric acid cycle, it is going to produce GTP rather than the ATP as in other tissues, what we have seen in other reactions. And this GTP is going to be used up for the reaction of this conversion of oxaloacetate to phosphoenol pyruvate, catalyzed by this phosphoenol pyruvate carboxykinase enzyme. So it is showing that it is providing a link between the citric acid cycle activity and the gluconeogenesis process. So it is showing exact link where the citric acid cycle activity and the gluconeogenesis activity which is happening, right? So it prevents and as well as in order to prevent the excessive removal of oxaloacetate for the gluconeogenesis process. It also shows the prevention of the excessive removal of the oxaloacetate for the gluconeogenesis, which would impair the citric acid cycle activity, right? So if you remove more amount of the oxaloacetate for the gluconeogenesis process to carry out, so it is going to impair the citric acid cycle activity. So that's why it has to be converted oxaloacetate to phosphoenol pyruvate some amount. Then it is continuing your gluconeogenesis process. So this phosphoenol pyruvate is getting converted back to the next product 2-phosphoglycerate, next to 3-phosphoglycerate, then 1,3-bisphosphoglycerate, then glycerolidate 3-phosphate, then fructose 1,6-bisphosphate, then fructose 6-phosphate, then again glucose 6-phosphate, then glucose 6-phosphate to glucose molecule. That is the synthesis of glucose that we will see in detail in the next PPT. Thank you.